welcome to day 3 of AHA's Scientific Sessions 2020. This is our daily recap. It's turned into a really fun event where we get to think about some of the great stuff we saw this past day and look ahead to tomorrow. I'm joined tonight, as usual, by AHA President, Dr. Mitch Elkind. Mitch, welcome.
Thank you.
Our Vice Chair, almost Chair for Scientific Sessions Program, Dr. Manesh Patel from Duke University.
Excited to be here.
And his colleague, Tracy Wang, Dr. Tracy Wang, who is professor at Duke as well, and the Director of Health Services and Outcomes Research. Sorry, Tracy, hope I said that right.
Hello, everyone. Thanks, Don.
Absolutely. So really terrific day. We had wonderful sessions, I thought, on diabetes management and heart failure management earlier today. And then, of course, a wonderful late breaker, fish oil, fancy drugs, and frustrations in lipid management. Tracy, you were a moderator for that session, and it has my favorite clinical trial. I must say it is the smallest clinical trial we're presenting this year, but I think in many ways the most intriguing, and talking about, of course, the Samson trial. Tell us a little bit about Samson and your thoughts about it.
Yeah, this was the little trial that can, I guess. This was a very creative trial, one that we've been waiting for. We clinicians have been waiting for a really long time. The trial took 60 participants and randomized them to alternating between a statin, a placebo, and an empty pill bottle, and basically used a very simple smartphone app to try to quantitate daily patient-reported symptoms. And so what they found was that the act of taking a pill in and of itself resulted in symptoms, but the difference between taking a statin and a placebo was quite small. In fact, most of 90% of the symptom burden was actually attributed to the nocebo effect. So that was really interesting. But more interesting is that this trial recruited patients who'd previously stopped their statin because they had experienced what they felt was side effects early after starting the statin. And after going through this experiment, these patients were followed up. And by virtue of learning that most of their symptoms might have just been because of the expectation of potential side effects, half of these patients, after the study was done, was able to resume statin therapy. And it'll be interesting to see if they were able to continue that long term. What I took from this as a clinician is that we really need to prep our patients better on how to start a statin, because in real life, unfortunately, I can't do a blinded, randomized N-of-1 trial for all of my statin naysayers. But I'm definitely thinking a little bit more about how to incorporate the information from this trial into the types of conversations I'm having with my patients. And it kind of reminds me of that really famous FDR quote: "The only thing we have to fear is fear itself," because I think that's basically what this trial is telling us. We know that the placebo effect works if people are expecting to feel better. Here it's kind of the opposite. The nocebo effect is really a problem because people are expecting to feel worse. And actually, I might, tap Mitch to explain this to me. Is there a neurobiologic mechanism for this nocebo effect? Your thoughts on this?
Sure. Well, those are great comments, Tracy. I think that was terrific. And I know that this is one of Don's favorite trials as well. And I do think that, yes, I would add that this study has important neurobiological effects as well as clinical cardiovascular implications. It's another great example of the intersection between cardiovascular science and brain health. I'm not an expert in the nocebo effect or placebo effect, but I can tell you that we're beginning to understand the brain pathways and the neurotransmitters that are involved in those responses. And it's clear that they're real. They're biological, and they can be manipulated as well. It involves the opioid system, the dopaminergic system, other neurotransmitters. We know that the prefrontal lobes are involved in the placebo-nocebo response. Those are, the parts of the brain that are responsible for executive functioning. And one interesting aside here is that people with dementia are actually less likely to have these kinds of effects, presumably because their brain is not influencing those mechanisms as much. But I would say that, regardless of the specific mechanisms, it's clear, as you said, that patient expectations can influence how drugs either work or cause side effects. Muscle pain from statins is a perfect example. And so from a clinical point of view, I think what it tells us is that the physician's words play an important role in the therapeutic response. And, we don't just give a drug. We never just give a drug, really. It's always being done in the context of the patient-physician relationship or dynamic. And similarly, a patient's psychological makeup can influence, how the response goes. Is a person anxious, for example, or are they fearful in some way? Have they had adverse effects before that might influence their response now? And I think it would be interesting to know, I didn't see this, but it would be interesting to know if they captured any of that in the Samson trial. But more research definitely needs to be done. But, I would say, Manesh, I think there's little placebo effect, as far as I know, as a neurologist, in left main disease. Perhaps, and I know there's a bit of controversy around that. Perhaps you could enlighten me on that.
Yeah, no, Mitch, it's a great question. In fact, I think, as I was thinking about this placebo conversation we're having, or nocebo conversation, I think a few years ago, this started as we started to do renal denervation. We had arms of sham procedures and then angioplasty by some of the same investigators where we tested that. The big controversy right now, I think, is discussed in the interventional sort of best of interventional sessions today, is ongoing conversations about the unprotected left main trials that happened now over a few years ago, the XL trial, which had both surgical and interventional colleagues doing randomized trial and reporting results. And a lot of the controversy revolves around the definition of myocardial infarction and how severe it was identified. And then a mortality signal seen late. Several trials, and what I think it highlighted for me is, again, the ongoing conversation that physicians and clinicians are having with their patients, because they want to know what's best for them. And they're not just interacting with a therapy, they're interacting with the team that has their own belief system. And so I would say right now, the state of the science for left main revascularization is still a heart team with plenty of information to tell us about the risks and benefits. With, I would argue that most of the patients are still likely getting thought of for bypass surgery, and an angioplasty for isolated left main can certainly be done, but that's an area of controversy even with these trials. Speaking of controversies, Don, again, in day 3 of the AHA, I didn't think I would say this. I've heard a lot about interventional controversy, left main versus PCI and cabbage, but what about fish oil and omega-3s, its strength and omemi? Tell me maybe what you think about that controversy with REDUCE-IT and the findings we saw today.
Yeah, well, our listeners will remember the REDUCE-IT trial presented at these meetings 2 years ago, an unexpectedly large benefit from a 4 gram per day dosing of icosapent ethyl, a fairly refined EPA dosage with no DHA. And then today, 2 trials presented, the strength trial and the omemi trial, in different patient populations, but both of those were sort of moderate dose DHA EPA combo pills, and both of them were null. And so we're left with this conundrum, what's going on here? And I think lots of great discussion today. There's certainly something about the placebo composition that we need to figure out. In Strength and Omemi, they used neutral corn oil placebo preparations. In REDUCE-IT, they used a mineral oil, which increased the inflammatory markers in the patients who got the placebo, increased the LDL cholesterol. The question is, did that make the drug look better because the placebo was worse? And I don't think we have fully the answer to that question. A lot of good basic science and animal studies suggesting that actually high-dose EPA really is the best way to go, and it does have mechanistic and biological plausibility as a viable way to think we can actually reduce atherosclerotic events with this high-dose refined EPA drug. However, the other thing that is clearly coming to the fore is a signal of adverse events, specifically much higher risk for atrial fibrillation with all these fish oil preparations. That may not be a trade-off we're willing to take, particularly if it's an expensive drug or it's a drug that only has marginal benefit. Bottom line for me, and Tracy, I'd love your thoughts about this, I think we need another trial. I think we need a trial with icosapent ethyl versus a corn oil placebo, and ideally also with a third arm that has a DHA EPA combo at a good high dose. But love your thoughts about that.
Lots of Twitter controversies and theories behind this. It's hard to really compare across trials when each of the trial is using a slightly different agent and the control groups are different, not to mention different populations. Even background statin therapy is different across these trials. So I think there's still a lot to sift through. I think one of the great things I've loved about these trials is that there are some great inflammatory and lipid biomarkers that are drawn here as well. So hopefully in between trials, we can start teasing out some of the mediating effects to really give us guidance. And don't forget, there's that atrial fibrillation, that pesky little thing that's rearing its head now that's making us all think, we're seeing this in all of these trials, and all three major trials now. What are we supposed to do with our patients who have paroxysmal AFib or who are at high risk of AFib? So lots here to digest.
No, absolutely right. Plenty more publications to be had here, I'm sure. Manesh, you've been kind of wandering around the meetings and have some great insights, I think, into all the other things, not just the late-breaking science, but all the other things that people need to make sure that they catch. Some thoughts about that?
Yeah, Don, I've said it for a couple of nights, and I really think, just like everything else, we're adopting and seeing new things as we do these virtual meetings. And one of the really exciting things is that we have the ability now on demand to go see posters and go review the Herrick lecture, which I understood, Clyde Yancey just gave is supposed to be amazing, and I want to go see that after this, or the structural racism talk, or the Young Investigator Award winners that you can see at the Heart Hub, at the Council Connects. Just seeing the names and the list, you can go see the presentations. I think one of the powerful things that'll probably be long lasting out of these sessions is that there's going to always now be some hybrid version. We really do miss the in-person connection for sure, but the ability to go find the content, to see that poster that you might not always have gotten to walk in front of, maybe see the comments others have had. And so I think from the Young Investigator Awards, the posters, and the ability to see this on demand into January is going to teach us, I think, the ability that this is going to move us forward in how we share this information. And honestly, it has increased the amount of different things I think all of us can see. And I think the conversations at least are hopefully going to be richer. So I think that's really interesting and something new. I'd be interested in what you see coming up in the next day or so, Don, because I think the ability to go through the entire program is always exciting now that we have this virtual format.
Yeah, thanks, Manesh. I think you're absolutely right. I've really enjoyed the opportunity to not just see the late-breaking science live, but then circle back, the things that were happening at the same time that I can still watch, still sort of feel the excitement of being in the room where it happens. And then in between times, catching those on-demand things. And there are some amazing original science out there, as well as some really good main event and other invited presentations that summarize kind of the state of the clinical care and science right now. So this is a pretty remarkable platform, and I think we're gonna have this in perpetuity, which is a good thing. So
yeah, I think sorry to interrupt. I was just saying, I think we're gonna get to the Netflix world, right, where enough people are viewing things, and we're starting to see that, they know I like to see these things or those things, and we're going to present that to people.
Yeah, but I don't think we'll be seeing people say, hey, come on over, we'll do AHA and chill. Maybe, I don't know. Anyway, looking ahead to tomorrow, we have late-breaking session 6, which is entirely about atrial fibrillation, a bunch of different screening studies helping us understand how we might find occult atrial fibrillation, really great studies, as well as a treatment trial with early cryoablation versus antiarrhythmic drug therapy. It's Guideline Day, a number of sessions reviewing the recent ECC, Emergency Cardiovascular Care and CPR guidelines, the other guidelines that have been published this year by AHA and ACC. We also have the Frontiers in Thrombosis seminars, and another main event on artificial intelligence and machine learning and imaging, and how that's really revolutionizing our ability to read studies.